Okay, we're getting ready to reconvene the afternoon session. My name is Chuck Haas from Drexel University. I'm one of the committee members, and we have one speaker face-to-face and another coming in remote. And as with the earlier sessions, we'll hear from both of the speakers and then take the questions for both of them as a group at the end. So our first speaker is Jennifer Martiny here from the University of California, Irvine.
Thank you, everybody, for coming. And it's an honor to be here today. Very easy trip for me over from main campus. And I'd like to talk to you today about microbial biogeography, which is an area that my lab has done a lot of work in for the last decade and a half. And the way we've been thinking about microbial biogeography patterns these days is in terms of in light of traits, as Jay and others have mentioned this morning. And so you may not think of yourselves as a biogeographer, but if you study community variation and look at any plots such as this, where you're looking at the often the taxonomic variation of a microbiome community, then we're often biogeographers then want to understand what are these patterns over space or time. What are the ecological and evolutionary processes that underlie these patterns? And ultimately, a lot of us are interested in biogeography to try to understand what these patterns mean for the functioning of a microbial community. And so, we've been doing this for quite some time, trying to document what the patterns are, what are the relationships with environmental variables. And we sort of were getting stuck at that point, and we wanted to go a little bit beyond just documenting these patterns and think about the processes underlying them. So we started to think about traits, which many people in this room have been thinking about for a long time. Also for plants and animals, ecologists have been thinking about how to interpret biogeography in terms of traits. So for microbes, we might think of, you can classify traits in many different ways, and one way I'm going to talk to you today about is thinking about traits in terms of a range of them from simple to complex traits. So on the one end of a simple trait, we might think about something like organic phosphate utilization. And so for this, you have to be able to produce an alkaline phosphatase, and that might only require one gene. A bacterium may only need to have one gene in order to produce this enzyme. And so that I would call a simple trait, because that trait might be picked up very easily, for instance, by horizontal gene transfer. And it may not affect many of the other processes going on in the cell. On the other extreme, you might think about something like salinity preference as a very complex trait. There's no particular gene or operon that we can look for that says this is the part of the genome that specifies what preferred salinity this organism likes. And so it's much harder to study. You imagine that many different traits and genes within the organism are involved in determining whether or not this organism is a marine versus freshwater organism. And so in thinking about traits, though, one thing as we want to relate them to taxonomic composition, we have to think about whether or not they're conserved. So I like to start by thinking about this idea that really, instead of a tree of life, we need to think about a web of life proposed by Ford Doolittle some time ago. And so his point was that maybe we might not know very much. The taxonomy of a microbe, especially a bacterium or archaean, might not tell us very much about the traits of any one organism. And that's because there would be so much horizontal gene transfer, as well as perhaps gene loss and rapid evolution among microbes, that their taxonomy really doesn't tell us anything about that organism. And so we've been thinking about this for quite some time. And so just to put this into a context I'm going to talk about a little bit here in terms of phylogeny, if there's so much horizontal gene transfer and rapid evolution that it really breaks up any vertical inheritance, then we'd expect any particular trait. Here I'm just plotting, a trait that might confer, that you might carry or not carry. It might be randomly distributed across the phylogeny. In contrast, if there is a phylogenetic signal, then we'd expect to see clustering across the tree, whereby you have similar traits within one clade versus another clade. And so this doesn't mean that there isn't any horizontal gene transfer, it just means that perhaps there's not enough in order to break up this vertically inherited signal. And so a lot of groups have been thinking about this and trying to determine whether or not traits are conserved and what types of traits. And so I'm going to review just give you a couple of examples, especially from our large microbial ecology group here at UCI. And so a few years ago, Adam Martiny and Kathleen Treseder, two faculty here, they just looked at whole genomes of bacteria and archaea, and they said, let's take these genomes. We know this is just potential traits, but let's look at the genomes and pull out what types of traits we can from those genomes. And they asked they recorded the presence or absence in these genomes across the phylogeny. And here I'm just plotting 3 different types of traits, and you can see that the presence of these traits are not randomly distributed across the tree. They are definitely conserved to some extent. And so you can do that for metabolic traits, and overall they found of all the traits they looked at, I believe it was somewhere around 97 traits, they found 93% of them were significantly clustered, and so not randomly distributed. And other people have used biogeographic patterns or distribution data to look at this question as well, and here I'm going to talk about a project where it's actually from an experiment nearby at UCI, about 30 minutes away in our hills, where Mike Woolden has been manipulating precipitation, so similar to the story that Sarah was talking about today. So we were just looking at the microbes in these plots in ambient versus drought-treated plots and asked whether or not there was a conservation of the response of those organisms. And so we were looking at both bacteria and fungi and saw similar results. This is the tree for the fungi that were present. And if the node is coded in blue, then those are fungi that responded positively to drought, and those in orange and red responded negatively. And so what you can see is that, again, these nodes are highly clustered around the tree. And so it seems that the response trait, or the group of traits that are responsible for the response to drought, also seem to be clustered, even though this is probably a very complex trait. And so in addition to just saying that, trying to figure out whether or not traits are conserved or not, we also have been thinking a lot about how deeply are traits conserved. And so just to compare, then you might have a trait that's very shallowly conserved, and so may have a random or very slight phylogenetic signal, all the way up to some that might be very deeply conserved and maybe only evolved once across the tree of life. So we just, together with Jay Lennon, who you heard from this morning, and Adam Martiny and Stuart Jones, we reviewed the literature recently to look at, of the studies that have looked for trait conservation, did we see variation in how deeply those traits were conserved? And so yes, we did indeed find that most traits do seem to be conserved, and not only are they conserved, but I just want to point out that this average here in the red line, that they're significantly differently conserved depending on the trait that you look at. And so we speculate then that maybe there's some sort of hierarchy of trait conservation, that maybe there's a predictable pattern of what types of traits might be less conserved than others. And we speculate that that might be related to the complexity of the trait. So again, the ones you'll notice up here that are the traits that are most deeply conserved are methanogenesis and pH preference. And so those appear to be quite deeply conserved, and that would make sense if those traits are encoded by many different pathways within an organism, whereas something, again, like phosphorus acquisition turned out down here where we expected in terms of its simplicity. Okay, so then I was asked to talk about sort of how we might think about making predictions, and so I'll go back to a prediction in terms of biogeography. And so if I'm comparing in this cartoon 2 different traits, one carbon substrate use that we think is shallowly conserved, and so there I'm going to use hatch symbols and solid symbols and that may be conserved perhaps just at the genus level or even at a finer scale than that. In contrast, something like pH, if we plot that on our cartoon here, if I color code it in orange and blue, that might be conserved down at the phylum level. So what I predict then, if I go out to a landscape and are sampling different communities, would be that I would see different shifts in my taxonomy depending on what gradient I'm looking at. So let's take the y-axis here, the pH gradient. And so if I'm sampling across a landscape where pH is turning over and changing across that landscape, I would expect then to see a huge shift in the phylum level composition of my community. And so there's many more blue cells here in this sample than in this sample down here. In contrast, on the x-axis, I'm drawing what I would expect would happen if we start looking across a range of different types of carbon substrates. And so if the organisms are not so conserved in terms of which carbon substrates they can use, then we'd only see a very fine scale at the fine scale resolution where we would see a turnover in the genera or species level organisms once we cross the carbon substrate. The carbon substrate gradient. And I just want to give you an example of where we think that we actually do see that there is some evidence for this sort of hierarchy of trait conservation. And I'm going to tell you about Procholorococcus, which is a marine bacterium, but is one of the best studied free-living microbes that we've looked at. Not me personally, but many people. And so this is sort of the the typical macroevolutionary picture that we have about Procholorococcus. And so I mentioned it's a marine organism, and so there's a very deep division of the group where some organisms in this are classified in this highlight clade, and they prefer to be at the surface of the ocean. And then there's others that prefer to be deeper down in the low light clade. And then within this highlight clade, you can divide up the clades separately by whether or not they prefer high iron regions of the ocean or low iron regions, and so forth, down to where you're talking about whether they prefer high nutrients or low nutrient environments in terms of the types of phosphorus and nitrogen that they can take up. That's very finely conserved. And this agrees very well with what is seen in biogeographic patterns. So again, using an example from Adam Martiny, he looked at he varied the taxonomic resolution at which he was looking at his samples of Procholorococcus. And so he was using the ITS region, which was able to get very fine scale resolution. And only at the finest scales could he see that nutrients were the most important in explaining the variation across his samples. But if he clustered those OTUs, those taxonomic units, at a broader and broader level, eventually got to the place where none of the other environmental characteristics could explain compositional variation, but just except for light, which is explaining which happens to be the trait at that level that it differs across the clades. And so I think there are a couple of interesting implications if this trait hierarchy holds. And the first is that biogeographic patterns observed will depend on your genetic resolution that you look at. And that probably isn't surprising to any of you that have been doing work on microbiomes, but I think it's interesting to think, and it's actually useful to think of a reason, the mechanistic reason why that would be the case. So if a student now hands me an NMDS plot and he sees this variation, I'm always asking them, okay, well, what is the trait underlying those differences across your samples? Why do you think they're diverging? And why did you classify them at the 97% level or the phylum level or whatever classification they used? And the second implication is that the taxonomic or genetic level that you use to look at your pattern then provides a hypothesis about the traits under selection. So you can go to a different environment, and if this hierarchy holds up across the whole microbial tree of life, then even if we're moving across systems, we might be able to test hypotheses, whether you're looking in an ocean or in a built environment. So I'll just end there and take questions after our next speaker. Thank you.